The mushroom that ate Chernobyl and what it's building there now. You are not supposed to be here. The signs say so, the fences say so, the dosimeters that click faster the closer you get say so. The empty apartment blocks, the rusting fairground wheel, the schoolroom still scattered with books that no child has touched since 1986, all of it says so. But something is here, something that was not here before, something that arrived not despite the radiation but because of it. And it is building. On April 26, 1986, reactor number 4 at the Chernobyl nuclear power plant exploded. The blast released 400 times more radiation than the bomb dropped on Hiroshima. A plume of radioactive material spread across Europe. The Soviet government evacuated a zone of roughly 2,600 square kilometers around the plant, relocating over 350,000 people, and declared it uninhabitable. They were not wrong to do so. The radiation levels in the immediate exclusion zone were lethal. The landscape, by every measure science had for evaluating biological viability, was a dead zone. Within weeks of the explosion, the trees in the forest closest to the reactor turned a deep rust-brown color and died. Scientists named this area the Red Forest. The soil, the water, the air, contaminated. The region was expected to remain biologically devastated for decades, perhaps centuries. What actually happened was stranger than anyone predicted. Within a few years of the evacuation, wildlife began returning to the exclusion zone. Wolves, lynx, bears, wild horses, populations of animals that had been hunted nearly to extinction elsewhere in Europe were thriving inside the most radioactive landscape on the continent. Ecologists studying the zone found that despite measurably elevated radiation levels, the absence of human activity was, for most species, more beneficial than the radiation was harmful. But the truly unexpected arrivals were not the wolves or the horses. They were the fungi. In 1991, 5 years after the explosion, researchers studying the reactor discovered something that rewrote their understanding of what life was capable of. Growing directly on the walls of the damaged reactor itself, inside the structure, in the most intensely radioactive environment on the surface of the Earth, were black fungi. Not surviving there, not clinging to existence at the margins, thriving. And growing toward the radiation source. The fungi were identified as belonging to several species including Cladosporium sphaerispermum and Wangiella dermatitides, both members of a class called radiotrophic fungi. Radiotrophic. The word means, with a precision that still unsettles biologists who work with it, radiation-eating. These fungi contain extraordinarily high concentrations of melanin, the same pigment that gives human skin its color and protects it from ultraviolet radiation. In radiotrophic fungi, melanin is not a protective mechanism. It is a harvesting mechanism. Ekaterina Dadachova, a radiobiologist at Yeshiva University, published research in 2007 that demonstrated what was happening inside these fungi. They were using ionizing radiation, the same radiation that destroys DNA, causes cancer, and kills most living things, as an energy source. The way plants use sunlight, the way your cells use glucose. The fungi were eating the radiation and using it to grow. Dadachova's paper sent a quiet shock through the biology community. Photosynthesis, the process by which plants convert light into chemical energy, had been considered one of the foundational innovations of complex life. Now here was evidence of a completely different mechanism, radiosynthesis. A way of harvesting energy from ionizing radiation that no one had suspected existed, performed by organisms that had been living alongside humans for the entire history of the species without anyone noticing what they were capable of. The reactor at Chernobyl had not killed the fungi, it had revealed them. But the fungi were not just eating the radiation. They were doing something with it that has implications far beyond Chernobyl. They were breaking it down. Not metaphorically, not slowly. In measurable, documentable, peer-reviewed ways, the radiotrophic fungi at Chernobyl were metabolizing radioactive material and incorporating it into their biological processes, reducing the concentration of certain radioactive isotopes in the areas they colonized. Researchers including Arturo Casadevall at Johns Hopkins University have spent years studying what the Chernobyl fungi are doing to the reactor site at a biological level. What they have found is that the fungi are not simply surviving in the contaminated environment. They are remediating it, processing it, slowly, patiently, through the same enzymatic machinery they use to break down any organic material, digesting the radioactive contamination and building something new from its components. This process is called mycoremediation, And Chernobyl is the largest unintentional experiment in mycoremediation in human history. Paul Stamets, the mycologist who has spent 5 decades documenting what fungi are capable of, has argued that the Chernobyl fungi represent one of the most important biological discoveries of the 20th century, not because of what they tell us about Chernobyl, but because of what they tell us about fungi. Every ecosystem on Earth has been contaminated by something: oil spills, industrial runoff, agricultural chemicals, heavy metals, radioactive waste. The list of substances that human industrial civilization has introduced into the biosphere and that no existing technology can economically remove from it is very long. Stamets' argument, supported by decades of laboratory and field research, is that fungi have been solving versions of these problems for 450 million years. Long before there was oil, there were hydrocarbons, and fungi evolved to break them down. Long before there was industrial mercury, there were heavy metals in volcanic soil, and fungi evolved to process them. Long before there was a reactor at Chernobyl, there was background ionizing radiation from cosmic rays and radioactive minerals, and fungi evolved to tolerate and eventually harvest it. The contamination is not new to them, only the scale is new. In 2019, researchers sent a sample of Cladosporium sphaerispermum, one of the Chernobyl fungi, to the International Space Station. The purpose of the experiment was to test whether the fungi's radiation harvesting abilities could work in the context of deep space travel, where astronauts are exposed to cosmic radiation levels far beyond what Earth's atmosphere filters out. The results, published in 2020, were significant. The fungi grew on the ISS, demonstrated measurable radiation shielding properties, and survived conditions no other organism used in space biology experiments had previously tolerated. The mushroom that ate Chernobyl is now being studied as a potential shield for the first human missions to Mars. Here is what is happening right now in the exclusion zone while the fences stand and the signs warn you away and the dosimeters click. The fungi are still growing. The red forest, which was stripped of life so completely that scientists used it as a baseline for what total biological devastation looks like, is recovering, slowly, patchily, but recovering. And the recovery is being built layer by layer on a foundation of fungal activity, mycelium threading through the irradiated soil, radiotrophic species metabolizing the contamination, saprophytic fungi breaking down the dead organic matter, the fallen radioactive trees, the collapsed structures, the abandoned materials of a civilization that evacuated in hours and left everything behind. The fungi are eating the ruins of Chernobyl, and from those ruins they are building soil. The same thing they have been doing without interruption since long before the first human being walked the earth. The same thing they will be doing long after the last human structure has crumbled and been reclaimed. They do not know that Chernobyl was a disaster. They do not experience the exclusion zone as a dead zone. For them, it is simply a resource. A very large, very energy-rich resource left untouched by the species that created it. There is a particular kind of humility that comes from learning what happened at Chernobyl, not the disaster but the aftermath. You built something so destructive that you had to abandon it. You evacuated a civilization from the landscape and declared it uninhabitable. And while you were gone, the fungi moved in and began, in their patient, distributed, 450 million-year-old way, to fix it. The question Chernobyl keeps asking is not whether the planet can recover from what we do to it. The evidence from the exclusion zone is clear: it can. It is, not on human timescales, not in ways that will benefit the people who were evacuated or their children. But the mycelial network does not work on human timescales, it never has. It is working right now in the dark, under the red trees in the reactor walls, building something.